Welcome to another episode of Interview with a Biologist. I'm your host, Marie Lucido, and I'm joined today by Dr. Jennifer Johnson, who is Assistant Professor of Plant Eco-Physiology in the Department of Ecology and Evolutionary Biology from the University of Kansas, and Dr. Karine Prado, a research specialist in plant biology at Michigan State University. How are you both today?
I'm delighted to be here with you today. Thank you so much for the invitation, Maria.
Thank you for your interest in your research, and thank you for inviting us to this podcast. It's truly appreciated.
Of course. Thank you very much. I appreciate you taking the time. We'll start with Dr. Johnson, and then we'll move to Dr. Prado. But in general, would you mind telling us about your research interests?
Of course. I would say I'm broadly interested in physiological processes, and particularly interested in photosynthesis and respiration. I work on projects that range from exploring how these processes work down at the molecular level up to projects that focus on how to monitor their aggregate activity at the scale of the whole planet. I'm most deeply interested in questions that relate to how the Earth's climate impacts photosynthesis at terrestrial plants, and also how photosynthesis feeds back to influence the large-scale functioning of the Earth system.
Well, I'm excited to dive deeper into that, so thank you. And then Dr. Prado, what do you like to research? What are your interests?
I'm interested in plant biology and in plant resilience to abiotic stresses, and more specifically, I study the mechanism of thermotolerance of thermophile to improve crop in response to rising temperatures.
And is there a particular crop that you focus on?
I work with multiple crops, and I have a side project on maize.
So, Dr. Johnson, as faculty here at KU, can you tell us a little bit about the classes you teach?
For sure. In the fall of 2025, I taught Biology 414, which is Principles of Ecology. And this semester, I'm teaching Biology 606, which is Ecological Plant Physiology.
For this particular paper, you both work together, but is there anything in particular that drives you as researchers?
Well, I would say that overall, my professional pathway has really been shaped by an interest in understanding how the Earth's carbon cycle and climate system work, and perhaps equally driven by an interest in contributing to the things that we need to do to make sure to keep the climate's temperature at a point that's safe for society. This relates to physiology because the carbon cycle is really driven by the process of photosynthesis, taking carbon dioxide out of the Earth's atmosphere and trapping it in an organic form. And then the process of respiration decomposing that organic carbon back into CO2 that's returned to the atmosphere.
Okay, what about you?
For me, key motivators for this include the desire to contribute new knowledge to the plant biology field and to make some beneficial impact on society. I dream to find some solutions to feed the global population, expecting to reach nearly 10 billion by 2050. And this global population increase will likely require finding solutions to increase food production despite challenging climates, limited land, water scarcity, and soil nutrient deprivation.
That is a great thing to help fight against because, you hear about now that there's food struggles and food insecurity. And with that many people on the planet, we appreciate you having scientists like you researching ways to try and stop it. Before we jump into your paper, I think it would be good for our listener to have a bit of a mental image of the plant that we're going to talk about. I know I'm a bit of a visual learner, so it would help me. Would you mind telling us the common name for the plant as well as give us a quick description of what it looks like?
The Destromia oblongifolia is known as Arizona honeysuit and is a perennial shrub native to Morave Desert and Death Valley. And this desert plant is exceptionally tolerant and thrives under temperature above 45 degrees Celsius.
So you recently published photosynthetic acclimation is a key contributor to exponential growth of a desert plant in Death Valley summer. What can you tell us about this research?
So it is one among many stresses that can disturb plant growth However, heat stress is often more damaging than other stresses because it directly attacks fundamental processes like photosynthesis and can cause irreversible cell damages and significant yield loss, especially when extreme temperature is sensitive growth stage like flowering and seed production. And a lot of regions may be facing issue with rising temperature, even if in considered cold region as Michigan. And our goal is to engineer heat resilient crops to have yield and production less sensitive to rising temperature and heat wave.
I could add a little bit about the way that Karine and I came to connect this unusually thermal tolerant plant that brought us together, Titostromia blangifolia, is one that became famous in plant physiology back in the 1970s. Researchers from the Carnegie Institution for Science had built what they called a mobile laboratory for studying photosynthesis of plants in natural environments. And they took this system to different kinds of contrasting habitats so that they could study the performance of plants in the places where they actually lived. And one of the places where they studied the photosynthetic performance of different plants was in Death Valley, California. When they went to Death Valley, they discovered that this one particular species had a thermal optimum for photosynthesis that was higher than any other higher plant in the world. And in a sense, this was exactly what they had been looking for, because Death Valley has a climate that is hotter than almost anywhere else in the world. It's the hottest place in the Western Hemisphere. And it makes sense that the evolutionary process has selected in that environment for plants that can be successful and that have combinations of traits that allow them to thrive under these extremely challenging conditions, very hot, very dry conditions. At the time that the Titostromia was discovered to have very thermotolerant photosynthesis, it was amazing, but it also was not clear exactly why and exactly how the plant managed to do this. And in many respects, the tools to answer those questions were just not available at the time that the researchers originally discovered this plant. And today we have many, many more powerful tools that are available to explore the mechanisms that control the performance of photosynthesis under extreme temperatures. And the idea of bringing modern tools to the challenge of understanding thermotolerance was really the idea that brought Karine and I together to collaborate on this project.
So we have a plant that is extremely heat tolerant, lives in a very, very difficult place for many things to survive. I guess, why is it so important that we research plants with high heat tolerance and in these extreme conditions when most of us are not living under that kind of pressure?
So it's a great candidate to decipher the fundamental molecular mechanisms and confer it resistance. And we can think about some impacts, and it can be the transfer of this mechanism into crops to improve their resilience to increasing temperatures.
Off the top of your head, do you know of any existing crops that have benefited from that kind of, genetic modification?
Is under progress, but so far they have no, except this study, they have no crop using the Titostromia mechanisms, but is in progress, and we are working on that just to try to transfer. For example, one good candidate in Michigan will be the sugar beet. So sugar beet is one of the top crop in Michigan, and sugar beet is very sensitive to heat. Even in Michigan, and even few degrees can be disastrous for the field and yield. And in the future, if sugar beet can be more tolerant to heat stress and heat wave, we can think just to maintain the yields, current yields. But so far, I don't think they have any group working just to improve crop with the knowledge based on Tilestromia.
There's always room for development. So fingers crossed that it ends up working out with sugar beets. And I just remember seeing, I think, a news story about how grapes originally, they had a very limited grow zone that ended up shifting more northwards, and that they're actually growing grapes in England now because of changing in climate and temperature. And so it would be interesting to see if the sugar beets, as they became more heat tolerant, if they had sort of shifts in their grow zones too.
I think that's really right on point. Mm-hmm. As the Earth's climate continues to warm, a lot of the crops that are currently growing very happily in particular places are likely to be challenged by the warming temperatures. If you look across all higher plants at the fundamental response of photosynthesis to temperature, you always see more or less the same general pattern. At very cold temperatures, photosynthesis goes very slowly. And as the temperature begins to warm, photosynthesis starts to go faster and faster and faster. It then will reach a maximum speed at some kind of intermediate temperature, which differs depending on the species you're talking about. But from that peak temperature where photosynthesis is optimized, as the temperatures keep going up, photosynthesis starts to decline. And the temperature where that decline starts is variable, but it's a universal decline that's observed in all higher plants. The basic concern when we look at the planet's future with a warming climate and an increasing human population is that we run the risk of essentially driving our current crops outside of the temperature range where they can really be maximally productive. So looking for a deeper understanding of the ways that evolution has solved the high temperature problem in other species and exploring strategies for bringing those evolutionary innovations into the crops that people rely on is a really timely problem. And it's relevant to almost every crop that we think about in terms of food or fuel or fiber production. Mm-hmm.
Yeah, I know we're currently seeing coffee beans and chocolate under some heat stress. So I'm sure many people would be happy to see them, better protected against these rising climate changes. Can you tell us about the methods you used to find your results?
Tilestromia omblogifolia is not a model plant yet, so we chose an holistic set of methods to decipher the mechanism underlying its resistance. Methods include photosynthesis measurement, macroscopy, gene expression analysis, and biochemistry.
When I was skimming through your paper, I saw some photos, I believe, of a lab setup that you were using. Can you tell us a little bit about what that was like?
Yeah, we use, I believe you are talking about the specific growth chamber mimic. Mm-hmm. It's a fantastic new growth chamber now we have at MSU. They can do at the same time high temperature, 47 degrees Celsius, and very high light, 2200 micromole ppfd. So it's very challenging to get this kind of chamber who can have the both together without any failure. And we are very happy just to get this fantastic chamber who can mimic exactly what's happened during a day in July. And just to get exactly what's happened, Jane modeled the climate in July in Death Valley, and we got this parameter in the chamber to mimic what's happened in the summer. And it's pretty hot, even if when you just open the door, you feel the bright light and the temperature inside.
During this study, what kind of findings did you have? And then what do those findings mean for further research?
Among main results, we can cite rapid growth under Death Valley condition. The plant tripled its biomass in just 10 days under extreme heat. with a photosynthetic optimum reaching 45 degrees Celsius. So it's one of the top described so far. And in response to Death Valley conditions, the plant also exhibits cellular change, such as relocation of mitochondria to chloroplast, reshaping of chloroplast in cup-shaped structure. The plant rewrites also the gene expression, including increasing expression of mini photosynthesis gene within just 1 day. And many are involved in protecting protein, membrane, and photosynthetic machinery from heat damage. And we also discover that the valley condition can boost the production of the ribisco activase. So it's a key enzyme in the control of ribisco, a crucial enzyme for photosynthesis. And for the impact of this research, so Tidestromia shows that plants have the capacity to adapt to extreme temperatures. And this survivor has still a lot of to teach us. But if we can translate this mechanism in crop, possibility Tidestromia may be transformative for agriculture facing challenge to grow at rising temperatures.
I might add just a little bit to complement what Karine has described, which is really to emphasize that the combination of different styles of measurements that have been brought together in this project is really unusual. It's understanding the response of pretty much any part of biology to temperature involves interactions that start in the genome, but are expressed all the way up into phenotype and performance. And in the essentially in the Tidestromia system, we now have characterization of genomic and transcriptomic and metabolomic and phenotypic and physiological observations that are nested and that provide a really unique opportunity to understand thermal adaptation in a holistic way that's really never been possible before. And I think it's, on the one hand, it's an amazing challenge because there are so many types of measurements to integrate, but it's also a really amazing opportunity that has incredibly powerful potential for being applied to the kinds of social objectives that Karine outlined. Mm-hmm. Karine, are there any of the avenues for future investigation that you would want to comment on, or what do you think?
For now, we start with this first paper. It was just describe the plant, and now we are focusing on specific protein, and hopefully for the long term we try just to transfer this mechanism in crop and just to prove any protein can be useful just to increase the resilience of this crop. So yeah, for now we have different projects, and also we have in this paper, we study a lot the leaf, but they have also a lot to learn from roots. So we are also starting to study the roots now, and they have very exciting results about how the root can be resistant to the heat stress.
That's awesome. I mean, it's so exciting when an avenue of research opens up different pathways. So I'm looking forward to hearing about what you get up to in the years to come. Thank you for joining us today, Doctors Johnson and Prado. I really appreciate you giving us the time and a deeper look into your research. And thank you for listening to Interview with a Biologist. You can check out the show notes for more information about the doctor's research, as well as a link to the research paper discussed in this episode. A full transcript will be available at biology.ku.edu.